right. Hello, class. Yes, good morning, good afternoon, good evening to my Histopath lecture classes. Okay. Yes, wake up, guys. So, thank you for dedicating some of your time to watch my videos in Histopath. Okay, so for this video, I will be discussing about staining, particularly the first portion of staining, okay? And that is on the different principles which are involved in the staining technique. And for this video, the last portion would be a discussion on your hematoxylin and eosin staining so that you would be able to understand the importance of the different reagents which are included in that particular staining technique. Okay, so it is something that you have to be very much familiar with. You have to know your hematoxylin and eosin staining technique by heart. Okay, ayan. All right, so let's start our discussion with the definition, of course, of staining. So what is staining? Okay, so staining, the simplest term or the simplest definition that we can give for this particular term is that this is a step or a process that involves the application of dyes on your tissue sections. Okay, so what is the purpose of staining? Staining, class, allows us to study the different architectural patterns of your tissue sample. Okay, and apart from that, take note that your staining allows optical differentiation so that the pathologist later on would be able to recognize and identify, okay, the different tissue components or constituents that are present in your sections, particularly those tissue components or constituents which are significant in the examination of your tissue sections. Okay, yes. Without staining, it will be very difficult for our pathologist, okay, to recognize or to identify, okay, the different tissue constituents that are present in our sections. Okay, so in order to do something about it, we have to stain our tissue sections prior to, okay, prior to examination. Ayan. So basically, there is another term that is also related to staining, and that is your impregnation. Okay, impregnation. So class, may I just emphasize that the impregnation, that we are talking about in staining is different from impregnation or infiltration. Okay, so when we say impregnation, all right, when we say impregnation in relation to histopath, okay, so in relation to staining specifically, this involves a different mechanism as compared to staining. Okay, so impregnation, so this involves the precipitation of heavy metals, okay, on certain components of your tissue section. Okay, so the key word there is precipitation. Yes, the keyword there is precipitation. So that means, okay, when your heavy metals precipitate on certain components of your tissue sample, they are not actually incorporated, okay, inside your components. They merely undergo the process of precipitation. Okay, so examples of reagents that we can use for precipitation include, we have silver staining. Okay, silver staining, this is a technique which is under impregnation. Yes, silver staining is a technique that is under impregnation. So, if you have encountered the term silver impregnation, okay, so that is actually a process that is similar to your staining. Why is it similar to your staining? Because in silver impregnation, okay, the components, okay, the component that is capable of precipitating your heavy metals would be the one which will have a color. Okay? Siya yung magkakaroon ng kulay. And take note, what is our goal in staining? To give color to the different tissue structures. And it's the same thing that that happens in your silver impregnation. So silver impregnation serves to give color to certain tissue components. Okay? So in particular, your silver staining, this is utilized, okay, for the demonstration of nervous tissue bulbs. Okay, this is utilized for the demonstration of nervous tissue as well as reticulin fibers. Okay, as well as reticulin fibers. So again, impregnation is not the same as staining. We cannot consider it as under your staining, but it has the same goal as your staining, and that is to provide optical differentiation, okay, to provide optical differentiation by giving color to certain structures, okay? So we go back to your staining. So the general rule when it comes to the staining of your tissue sections is that, okay, we follow the rule on the pH, okay, the pH of your structures. So in particular, your acidic structures will have a greater affinity for to your basic dyes, and your basic structures have a greater affinity for your acidic dyes. Okay, so based on these general rules of staining, we can therefore say that your acidic structures, they are considered to be basophilic structures. Okay, on the other hand, your basic structures are considered to be acidophilic. Okay, they are considered to be acidophilic structures. All right, so we go back to how we recognize the definition of some terms. So the term basophilic, what does philic means again? What does philic mean? Sorry, again. So it means loving, right? Loving or having an affinity for something. So in this case, okay, in this case, your acidic structures have an affinity to your basic dyes. So that's why they are basophilic. They love your basic dyes. So in contrast, when we look at the definition of your acidophilic structures, again, philic means having an affinity for something. Okay, loving something. And what does your structures love in this case? It's your acidic dyes. Thus, the term acidophilic structures. Okay, so I hope nobody gets confused, ha? Okay, I hope nobody gets confused with this particular general rules on staining. So in order for you to imagine what I am talking about, okay, so here is, okay, here are images of the reactions, okay, particularly of your eosinophils and your basophils, okay? So your eosinophil, yes, that's the reason why we call them your eosinophil. Why? Because they have, okay, they have an affinity for your eosin. And take note, class, that eosin is, yes, very good. That eosin is an acidic dye. Okay, so eosinophil, actually this comes from the term, or this comes from the fact that the eosin granules, your eosin, the granules rather, the granules of your eosinophil has an affinity, or have an affinity, okay, have an affinity to eosin. And again, just like what I have mentioned, eosin is an example of an acidic dye. Okay, so this is an example of an acidic dye. So therefore, what can you say about your eosinophil granules? Your eosinophil granules are therefore They are basic structures. Okay, so they are basic structures having a greater affinity for the acidic dye eosin. Okay, so that's why if you're going to look at the color, so the color leans towards your red, red or orange color. Okay, which is basically your acidic color. Okay, so in contrast, if we are going to look at the staining reaction of your basophil. So again, the reason why we call them your basophil because the granules of your basophil have, okay, an affinity to your methylene blue. Okay, so to your methylene blue. Okay, and again, what is methylene blue? Methylene blue is an example of a basic dye. Okay, so therefore, what can we say about the granules of your basophil? We can categorize them as acidic structures. Okay, so we can categorize them as your acidic structures. So that's why if you are going to look at your basophil granules, so they are leaning towards the basic color, which is blue or purple. Okay, blue or dark purple. Alright, ayan. So I hope everything is clear. Okay, so now let us move on to the three types. Okay, three general types of staining. Three general principles that govern the different staining techniques that we perform in histopath. Okay, so there are three. Ulit-ulit, ma'am. Okay. Ayan. So there are three general types. So first, we have your histological staining or histologic staining, histochemical staining, and then lastly, we have your immunohistochemical staining. Okay. So your histologic or histological staining, okay, so this involves a direct interaction or an interaction between your structure and the dye itself. Okay, so there is an interaction between the structure and the dye itself. Okay, so for your histological staining or histologic staining, this one follows the general rule. Okay, so this one is based on the general rules of staining, in which, again, your acidic structures will have an affinity to your basic dyes. Okay, and your basic structures will have an affinity to your acidic dyes. Okay, so it's as simple as that, my class and my lecture classes in histopath. Okay, so histological staining follows the general rules of staining. Okay, yes. So good examples of your histological staining include the different microanatomic stains that we have in the laboratory. Okay, others would include the bacterial stains that we use to color your bacteria, and of course, certain specific tissue stains. Okay, so that's under your histologic staining. There is an interaction between, okay, between your structures and the dye itself. All right. So now let us move on to your histochemical staining. Okay, so for your histochemical staining, in order for you to create color or optical differentiation, a particular reaction has to take place. Okay, yes. So that's why the simplest definition that we can give to your histochemical staining is that this is a staining, a type of staining that involves a reaction. So without the reaction, there will be no coloration. Okay, there will be no optical differentiation of the different structures. Right? What are examples of histochemical staining? So you have probably encountered this in your other professional subjects. Okay? So a good example of a histochemical staining is your Pearl's Prussian blue. Okay? Pearl's Prussian blue. All right? Yes. So your Pearl's Prussian blue, okay, this is a specific stain for your ferric. Okay, this is a specific stain for your ferric iron. All right, so again, what is ferric iron? Remember that the storage form of iron inside the body is in the ferric form. Okay, it is in the ferric form. All right, so basically, what happens is that the ferric iron that serves as the storage form of iron inside the body will combine, or it will react with one of the reagents that is incorporated in your Pearl's Prussian blue. And what is this reagent? We have your potassium ferri. Okay, so we have your potassium ferricyanide. Cyanide. Okay, yes. So your potassium ferricyanide, once it reacts with the ferric iron that is present, okay, that is present in your tissue sample, sige, ano natin, let us just try to label. So remember, your ferric iron is present in your tissue sample. Okay? Potassium ferricyanide, this is present as a reagent. Okay? It is actually the reactant that is incorporated in your Pearl's Prussian blue technique. Okay? So once the reaction takes place, so this will now result to the formation of your ferrocyanide. Okay, so this will now result to the formation of your ferric ferrocyanide, which is an insoluble blue pigment. Okay, which is an insoluble blue pigment. Right? So that's why the positive result in your Pearl's Prussian blue is blue. Okay, kaya nga, that's why it is referred to as your Pearl's Prussian blue, because the positive result is blue color. Okay, so without this reaction between your ferric iron and your potassium ferricyanide, without this reaction, your Pearl's Prussian blue will not be able to give optical differentiation for your ferric ions. Okay, ayan. All right, another example of your histochemical staining. What is another example? You have probably encountered this in your mycology, okay? Because it is one of the stains that we use in order to demonstrate fungi, okay? In order to demonstrate fungi, okay? What is this? We have your periodic, okay? Walang magsasabi ng periodic, ha? So this is periodic acid shift. Okay? Yes. So what specific reaction has to take place or is involved rather in past staining? Okay, so this is again a histochemical staining which involves a reaction between your periodic acid, okay, periodic acid plus, yes, plus the aldehyde functional group that is present in your carbohydrates. Okay, remember that the capsule, tama ba ako? Yes, remember that the capsule of your fungi, it is made up of carbohydrates. Okay, it is made up of carbohydrates. That's why your PAS is able to demonstrate your fungi. Okay, so periodic acid plus the aldehyde components of your, okay, I'm sorry, plus the aldehyde component of your carbohydrates, okay, of your carbohydrates will now react with another reagent that's incorporated in your periodic acid shift, and that is your dye, sorry, your dye or your stain fusion. Okay, so once this reaction among your periodic acid shift plus the aldehyde component of your carbohydrate plus the stain fusion, so this will now give a magenta color. Okay, a magenta color of the carbohydrate itself. Okay, yes. So again, without the reaction of periodic acid shift, periodic acid of the aldehyde component that is present in your tissue, okay, or in your fungi, plus of course the reaction of this to your dye, which is your fusin, there will be no optical differentiation. So color will not be able or will not be produced. Okay, ayan. So again, your pearl Prussian blue and then your periodic acid shift, these are examples of histochemical staining. There has to be a reaction that should take place in order for you to create optical differentiation or coloration in your tissue sections. Okay, ano pa ba yung, what are other examples? You have probably encountered this also in your histology. We have your Turnbull, Turnbull blue. Okay, yes, it is also utilized to determine the presence of iron deposits inside the body. Okay, but your Turnbull, okay, Turnbull reaction, it serves to differentiate or it serves to demonstrate your ferrous form of iron. Okay, Turnbull blue. Yes, if I'm not mistaken. Okay, ayan. So those are examples of your histochemical staining. So another good example of your histochemical staining would be enzyme histochemistry. Okay, so in enzyme histochemistry, again, your reaction has to take place. Okay, so in this case, we are detecting for the presence of certain enzymes in your tissue sections. So you learned this in your CC2. So if you are detecting enzymes which are present in your tissue sections, okay, in order to know that your enzymes are present, you have to give the substrate. Okay, the substrate that is or that will be acted upon if the enzyme is present in your tissue sections. Okay, so you have to provide the substrate. Okay, therefore, the reagent, okay, therefore, the reagent that is utilized for your enzyme histochemistry is basically the substrate. Okay, yes, yes, it is basically the substrate. Okay, so that is the active reagent that is incorporated in your staining technique. All right. So once your enzyme catalyzes, okay, catalyzes the reaction, okay, or once your enzyme acts upon the substrate that you have provided, so there will be the formation of your product. Okay, and of course, the formation of this product will now create a colored, a color reaction, a color reaction, thereby, okay, providing optical differentiation of some, yes, some components of your tissue sample. Right? So say for example, the enzyme is not present in your tissue. Then will there be a formation of a product? No. There will be no product or products for that matter. And so will there be optical differentiation? No. None pala. Will there be optical differentiation? There will be none. Okay, because your enzyme is not present. Right? Yes. So remember, your substrate will only be converted to your product once the enzyme is present in your tissues. So again, this involves an enzymatic reaction to be specific about it. And so enzyme histochemistry is under histochemical staining. Okay? The last one, but let me just make paalam, so I will just erase that. Okay? The last type of staining, we have your immunohistochemical staining. Okay, immunohistochemical staining. So class, whenever you encounter the term or the prefix immuno, always remember that this reaction involves a reaction or an interaction between your antigen, okay, it is an antigen antibody, okay, it is an antigen antibody reaction. All right, yes. So again, we have to identify which one is your antigen, which one is your antibody. So yes, what are antigens again from your immunology and serology class? So antigens, these are phenotypic markers. Okay, these are unique markers that are present on the surface, or it could also be present inside the different structures or the different cells which are present in your tissue section. Okay? So since they are phenotypic markers, they are unique markers which can only be found, okay, in certain structures of the body, and this now creates the specificity that is attributed to your immunohistochemical staining. Okay, so say for example, so that you would be able to understand. Say for example, if you are going to compare the two populations of lymphocytes. Yan, kasi ano na immunology and serology ang pinag-uusapan na natin. Okay, so if you are going to compare the phenotypic markers or the surface antigens present on the populations of your lymphocytes. Remember, there are two general populations of your lymphocytes. We have your CD4 positive, and then the other one is your CD8 positive, right? Okay. So since these surface markers are unique, you are then able to, okay, you are then able to provide color to a certain, okay, to a certain cell population or to a certain component of your tissue section. Okay? Yes. Kaya nga sabi ko, there is a high degree of specificity when it comes to your immunohistochemical staining. Why? Because antigens are unique. Okay? So in the case of histopathology, yes, we are after the reaction of your antibody, which serves as the reagent, to certain phenotypic markers which are associated with diseases. Okay, so say for example, malignancy or cancer. Okay, so depending upon the cell, okay, the cell or the structure that is involved in a particular malignancy, these malignant cells would have unique antigen markers. They would have unique phenotypic markers or surface markers. Okay, and so we are able to determine what kind of malignancy is present in the patient. Yan. So therefore, your antigen is the one that is being detected in your tissue. Okay? So the antigen nasa tissue po yan. Okay? And the antibody, this is the one that you are going to provide, depending upon, of course, the specific immunohistochemical staining technique that you are going to use. So the antibody, that is your reagent. Okay? And since there is a need for you to label your antibody, so there are several labels that we can use. Okay? So we can make use of, yes, Kasi your antibody, it has to be labeled, okay? In order for us to see that there is really an antigen-antibody reaction. Okay, so what are some of the labels that we use? So we can make use of your monoclonal, okay? We can make use of your monoclonal and polyclonal labels, okay? Or antibodies. Yes, labels. Ayan. We can also make use of your fluorescent labeled antibodies. Okay? Fluorescent labeled antibodies. And then we can also make use of your enzyme. Yes, enzyme labeled antibodies. Alright, yes. I'm sure you are very much familiar with these different labels because these are also being discussed in your immunology and serology. Alright, yes. So again, what is the function of your labels? Your labels serves as serves as indicators that really an antigen and antibody reaction has taken place. Okay, ayan. All right, so I hope the definitions of these three types of staining are clear to you. Okay, ayan. All right. So now let us move on to the different methods of staining. Okay, yes. So there are actually a lot of methods of staining which we can perform in the laboratory. Okay, so the first one, we have direct staining, okay, and then followed by indirect staining. So, sorry class, arranged it so that we would be able to differentiate, okay, differentiate the different methods of staining. All right, so when it comes to your direct staining, it has to be differentiated with that of your indirect staining, okay? So when we say direct staining, okay, when we say direct staining, this involves, okay, a direct interaction between your tissue and the dye itself. Okay, so say for example, this is your tissue. Okay, so say for example, ito po yung this is our tissue. Alright, yes, so that's our tissue sample. Okay, so in direct staining, so again, just like what I have mentioned, there is a direct, kaya nga siya direct, there is a direct interaction between your tissue and the dye itself. Okay, so say for example, this is your dye or your stain. Alright, so magkadikit sila. Ano pa ba ang ibang, what are other terms apart from direct? Okay, basta, there is a direct interaction between your tissue and the dye itself. Okay, yes. So basically, examples of direct staining involves giving color using the different aqueous or alcoholic dyes that we have. Okay, so say for example, that's your tissue, and then we have here your dye. Okay, so there is a direct interaction between them. So this dye, it can be in the form of an aqueous dye or an alcoholic dye. Okay, so another, we have your indirect staining. Okay, so in indirect staining, there is a need for you to make use of an additional reagent which we refer to as your mordant. Okay, why is it indirect? Because, okay, because if this is your tissue sample, okay, in order to create color, there has to be a bridge that should link the tissue to your dye. Okay, so this is your dye now, right? So this link or this bridge that links, okay, that links your tissue to your dye, that is your mordant. Okay, ayan. So without your mordant, no color will be produced. You have to have your mordant. Okay, yes. So color is produced using a mordant. Your mordant, therefore, this is an essential. Okay, it is an essential component that has to be present in order to link okay, your tissue to your dye in indirect staining. All right, so examples of mordants that we have in the laboratory, we have your potassium alum, which is the mordant that is incorporated in your Ehrlich's hematoxylin. Another example of mordants would be your iron. Okay, iron that is incorporated in your Weigert's hematoxylin. Right? So your mordant class, this can actually be applied on the tissue in 3 instances, okay? In 3 instances or in 3 ways. So you can incorporate it in your tissue before staining. Yes, if you can still remember the post-mordanting step that we can perform after fixation. So that is before staining. Okay, you can also incorporate it in the staining technique itself. All right, or you can also incorporate it in the stain itself. So just like what I have mentioned, your potassium alum in your Ehrlich's hematoxylin, so that one is already incorporated in your stain. Another, iron in your Weigert's hematoxylin, so that is already incorporated in your stain. Okay, so those are the 3 ways by which we can incorporate your mordants in your or on your tissue sections. Right? So another important term that you have to remember when it comes to your indirect staining would be your accentuators. Okay, so we have your accentuators. Right? So what are accentuators? So your accentuators from the term itself, okay? So this serves to hasten, okay? Hasten the staining process or the speed of staining. Okay, so how does your accentuator hasten or makes your staining process faster? It serves to increase the staining power of the dye itself, okay? And also, it improves the selectivity of the dye. Okay, so I repeat, your accentuator, it hastens the speed of staining, it increases the staining power, and it makes your dye more selective. Okay, so if we are going to compare your accentuator to that of your mordant, it is essential. Your accentuator is not essential. Okay, it is not essential. So meaning, even if you don't have your accentuator, okay, staining will still take place. Yes, because it will only improve the staining technique or the dye itself. Nakuha? Yan. Kaya nga siya, that's why it is referred to as accentuator. It will improve, okay? It will increase the speed of staining, right? Yes, so it is not essential. Your mordant, it is essential. It has to be present. In the absence of a mordant, staining will not take place. In the absence of an accentuator, staining will still take place. Okay, ayan. So what are examples of accentuators which are incorporated in our stains? We have your potassium hydroxide in your Loeffler's Methylene Blue. Okay, that's one. Another, we also have your phenol in carbonthionine and in your carbonfuchsin. Fuchsin. Okay, phenol in carbonthionine and in your carbonfuchsin. Okay, so those are examples of accentuators. All right, so I hope the difference between direct and indirect staining is clear. So direct staining, direct interaction, no need for mordant. Okay, indirect staining, indirect meaning there is no direct interaction between your tissue and your dye. Why? Because you need to have your mordant. Okay, yes. And then we also have your accentuator, which may be incorporated in our stains, whether it be direct or indirect. Okay, so your accentuators, they are not essential, but they can improve, okay, the staining process as well as the staining quality. Right? So now let us move on to another um, methods or other methods of staining. We have your progressive staining as compared to your regressive staining. Okay? So for your progressive staining, yes, for your progressive staining, this involves staining in a definite sequence and specific periods of time until the desired color is achieved. Okay? As compared to when we look at the definition of your regressive staining. So in regressive staining, your tissue sections are overstained and decolorized. So therefore, therefore, the main difference between your progressive staining and your regressive staining is the presence or absence of your decolorization. Okay, so when we say decolorization, this is otherwise referred to as differentiation. Okay, this is otherwise referred to as your differentiation. So in your progressive staining, no decolorization or differentiation is involved. Okay? Why? Kasi ganun siya. Okay, sorry. Okay. So, progressive staining, it does not involve decolorization or differentiation. You are merely going to immerse your tissue section until such time that you have achieved the desired color. Okay, until such time that you have achieved the desired color. Okay, so between your progressive staining and your regressive staining, progressive staining is less preferred. Okay, it is less preferred. Why? Because it's actually very difficult to achieve sufficient intensity or sufficient color intensity without coloring the other parts of your tissue section. Okay? So say for example, class. Example lang ha. Okay. So say for example po, this is one structure, okay? So that is one structure in your in your tissue section, okay? And then of course, apart from that structure, you will also have structures which are found beside or around this particular structure. So say for example, it is surrounded by these structures. Okay, ayan. Right. So that you would be able to understand. Okay. So what happens in progressive staining is that, okay, so if, for example, we only want this heart-shaped structure to be colored, okay, so you are going to immerse it, in your dye or your stain until such time that you have achieved, okay, until such time that you have achieved the color that you want. But what happens really in progressive staining is that, okay, so there is a very high probability that this heart-shaped structure will not be the only one that will be colored. Okay, yes. So the other parts which are not supposed to be colored will likewise be colored. Okay, yes, that's the problem in your progressive staining. Okay, even if you don't want these circular structures to be colored, they will be colored. Okay, they will still be colored. And of course, that will now affect the optical differentiation that we want to achieve in your tissue sections, right? So instead of coloring just this portion, the problem is your stain will leak, okay? It will leak or it will tend to color other portions of your tissue sections which are not supposed to be colored. Yes, that's the problem. Can you remove these colors in your other structure? No, you cannot remove it. Why? Because in progressive staining, there is no decolorization. Okay? There is no decolorization po. Right? So remember, what is decolorization? Selective removal of excess stain from your tissues, particularly those which are not supposed to be stained. Okay? So since there is no decolorization under the microscope, okay, your structures or several structures may have the same color. So that's the problem. So that's why progressive staining is less preferred. Okay? Um, since the other portions will be stained also, so this will now result in a diffused color. Okay? Diffused color, which will be more difficult for the pathologist to examine later on. Yes, and apart from that, some of the details might be obscured. Okay, like for example, for this one, do you think the pathologist will be able to know that this is the only portion of your tissue section that is significant? Okay, it will be difficult, even for your pathologist, to recognize structures which are really significant because of the diffused color. Okay, so that is for your progressive staining. Right, so now let us move on to your regressive staining. All right, so what happens in your regressive staining? So I will not erase that one, ha? Okay, so what happens in regressive staining is that your sections are overstained and again, they are decolorized. So the purpose of decolorization or differentiation is to remove the excess stain from your tissue, and apart from that, it also serves to remove the diffuse color which are not supposed to be present in certain structures. So say for example, okay, so in order for me to explain it further. So say for example, these are the structures that you want to color in your section. Okay, so in regressive staining, you are going to, sorry, okay, you are going to overstain. Okay, so since there is overstaining, you are sure, okay, you are sure that the desired color will be achieved. Okay, ayan. So, say for example, you are going to overstain it. Okay, and of course, in overstaining, the other structures, okay, the other structures will likewise be stained. Yes, so even this portion, all of this portion, okay, all of those structures that surround the significant structure, which is supposed to be viewed by your pathologist, all of those, okay, will have the same color. All right. The good thing about your regressive staining is that there is decolorization. Okay. So when you perform decolorization in regressive staining, what happens is that excess stain from your tissue and from the portions or structures which are not supposed to be colored will be removed. Okay, and so after decolorization, for example, of this particular tissue sections, what happens is that, well, okay, what happens is that, okay, only the structure that is supposed to be colored will be colored. Okay, so that's the good thing about regressive staining. Nakuha? Yes, the fact that you have to perform decolorization or differentiation makes regressive staining better as compared to your progressive staining. Okay? Yan. All right, so I hope it's clear. So the reagent that is used for decolorization or differentiation is referred to as your decolorizer or differentiator. Okay, so later when I will be discussing your hematoxylin and eosin technique, you will be able to see po or appreciate the role of your decolorizer and your differentiator, or rather your differentiator. All right. So other methods of staining, we have metachromatic, metachromatic staining and then orthochromatic staining. Okay, so they differ in the resulting color that is produced. Okay, so in your metachromatic staining, yes, just like what we observed in your mast cells and your basophils, the color that is retained is different from the original color of the dye. Okay, so for example, we have here these are examples of metachromatic stains. We have your methyl violet or crystal violet, Crescil blue, safranin, Bismarck brown, basic fuchsin. Sorry, wrong spelling yata yan. Okay, and then methylene blue, iodine, toluidine blue, azure, A, B, and C. Okay, so the color that is retained on your tissue section or by your tissue components is different from the original color of the dye. Okay, for your orthochromatic staining, on the other hand, so the color is retained. The color that is retained is the same, okay, to the original color of your dye. All right, so that's the difference between your metachromatic and your orthochromatic staining. Okay, so they differ in the color that is retained by your tissue components. All right, so another method of staining, we have counter staining. So when we say counterstaining, so this pertains to the application of a different stain to provide contrast and background. All right. So as for the different methods of staining, when we talk about your specific techniques, they are not exclusive, okay, to one method only. So like for example, your hematoxylin and eosin technique, okay, so it makes use of your regressive staining and your counter staining methods. Okay, so pwede one staining technique can have more than or can incorporate more than one method of staining. Okay. Yes. So what are examples of counter stains? So cytoplasmic counter stains, we have for us to have a red colorization of your cytoplasm, we can make use of your eosin Y, eosin D, and then floxin B. Okay. For yellow, we can make use of your picric acid, orange G, and then rose Bengal. And then for green, we have light green SF. Okay, and then we have lissamine green. So actually, it's yes, it's a little bit easy to, hindi naman very easy, but a little bit easy to remember. Okay, so for those stains that has a color in its name, so automatic, yun ang automatic that is the specific color that it produces. Okay, like for example, light green SF. Oh, what color does it produce? Green. Okay, lissamine green. Green again. All right, ayan. So another, we have for your nuclears, counter stains, we have for red, we have your neutral red, safranin O, carbine, and then we have your hematoxylin. Okay, for blue, on the other hand, we have your methylene blue, toluidine blue, and your celestine blue. So the easiest one actually to memorize would be the blue ones because they all have blue in their names. Yeah, so just be careful with hematoxylin. What else? Rose Bengal. Mm. Yan, baka we might think that, say for example, for Rose Bengal, so of course the first thing that comes to our mind is that it produces a red color, but actually it's yellow. Okay, for your cytoplasm. All right, so you have to know. Yes, you have to know your counter stains by heart. Yes, whether it is a cytoplasmic or a nuclear stain, and of course the specific color that it produces. All right, so other methods of staining. Yes, we have metallic impregnation. Yes, this is the one that I was talking about a while back. So again, when we say metallic impregnation, so this is a process in which certain tissue components are demonstrated not by stains, but, okay, it says here, by colorless, okay, by colorless solutions which are reduced by certain tissue components, okay? So upon the reduction of these colorless solutions, they will now produce an opaque. Okay, usually it is black. Yan. So usually it's black oak because it involves precipitation or reduction of your heavy metals. Okay? Yes, and this black precipitates or deposits will now adhere on the surface of your tissue. Okay? Whatever tissue component is capable of, yes, of reducing or precipitating your heavy metals. Okay? So therefore, if it involves precipitation only, okay, and the reduction reaction, this reagent, okay, the reduced form of the reagent is not absorbed by the tissue, but it is adsorbed. Okay? Adsorbed, not absorbed. So this now, the reduced heavy metal is adsorbed. So meaning it is held physically. Wait a minute. Okay, so it is merely held physically on the surface of your tissue components. Okay, that's the principle of adsorption. Okay, so it is held physically on the surface of your component as a precipitate or as a reduced heavy metal. Okay, ayan. So examples of solutions that we use for metallic impregnation include ammoniacal silver. Okay, we also have gold chloride. Yan. So particularly your ammoniacal silver, this is the one that we use to demonstrate your argentafin cells. So if you can still remember your argentafin cells, these are melanin producing cells, right? Yes, we have your argentafin cells, particularly the melanin. And then, of course, we can also make use of your ammoniacal silver for the demonstration of your intestinal glands. Okay, yes, argentafin cells also. All right, another colorless solution, we also have your gold chloride. Okay, yes, so that is for your metallic impregnation. So another method of staining, we have vital staining. So from the term itself, vital means it's alive. Okay, so this involves staining of living constituents. Yes, just like the one that we use in hematology for the demonstration of your reticulocytes. So that is an example of a vital staining. So in particular, it's supravital staining. But in your hematology, we don't usually specify it as a supravital stain. So vital stain is, if I'm not mistaken, sufficient to, yes, to describe or to classify the stains that we use for your reticulocytes. Okay, but in histopath, you have to know, okay, that it is specifically an example of a supravital stain. Okay, staining of living constituents. Examples, we have your trypan blue, okay, which is utilized for your reticuloendothelial cells. Janus green is utilized for the demonstration of mitochondria in living cells. Okay. Yes. So again, I would like to emphasize that the cells have to be viable. They have to be alive. Okay, because in vital staining, what we are trying to stain are actually the cytoplasmic components of your cell. Okay, yes. Because na naman. Why? Because a dead cell, okay, I'm sorry, a living cell, um, a viable cell, particularly the nucleus, okay, the nucleus of your viable cells is capable of resisting the stain. Okay, so how would you know that a cell is still alive if the nucleus is not demonstrated, if the nucleus does not have a color? Okay, so once the nucleus becomes demonstrated, okay, or if it is colored, it means that the cell is no longer viable. Okay, so it means that the cell is no longer viable. Okay, ayan. So because living cells, in general, they are capable of resisting okay, your stain. All right, so that's for your vital stain. Vital stain, they get incorporated in viable cells via the process of phagocytosis. Yes, so when these viable cells phagocytose the dye or the stain, of course, it gets incorporated in their cytoplasm. Thus, 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 the cytoplasmic structures become demonstrable. Okay, so there are two types of vital stains. We have your intravital staining, and then the other one is your supravital staining. Intravital staining, this is oftentimes used for imaging techniques. Like, for example, when you are going to undergo MRI, diba? MRI. So there is a need for us to incorporate, okay, your radiographic contrast dye. Okay, that's an example of an intravital staining. What else? In angiograms, diba? In angiograms, there is a need for you to incorporate or to administer rather your intravital stains. Okay, so in intravital staining, this involves administration of the dye into the body, and administration. It can be via three routes of administration. We have your intravital, IV, intraperitoneal, okay, or it could also be subcutaneous. Okay, so intravital, intraperitoneal, or subcutaneous. So what are examples of intravital stains? We have your lithium in the ink and then carmine. Okay, so we also have your supravital staining. So this this type of staining or vital staining involves administration of the dye after the removal of the tissue or cells from the body. Okay, just like the one that we perform for your reticulocytes in hematology. Okay, so that is an example of a supravital staining. Okay, so examples of supravital stains include, we have your trypan blue, toluidine blue, thionin, neutral red, Nile blue, and then lastly, we have your Janus green. Okay, so class, let me just finish your H&E technique. Yan, para matapos na natin yung part 1. Alright, so let's go to the last topic for this video. We have your hematoxylin and eosin technique. So just like what I have emphasized several times in my previous discussions, your hematoxylin and eosin technique, this is considered to be the routine staining technique that we perform in histopath. Okay, so if you are sleeping, wake up, wake up, wake up. This is the last portion already. Okay, wake up. Yes, you have to wake up for this portion because this is important. You have to know your H&E technique by heart. Okay, so a lot of questions do a lot of questions are asked in the board exam regarding your hematoxylin and eosin technique. And apart from that, this is one procedure that you have to know. Okay, you have to know in preparation for your internship also. Why? Because interns are the ones who perform your H&E technique. Okay, so what if your clinical instructor or supervisor asks you, what is the function of the two changes of xylene at the first part of your H&E? Okay. Oh, you don't know? Who's your instructor in Stupat? Oh, sabihin mo na. Okay. Ayan. All right. So wake up, wake up, wake up. Wake up, wake up, wake up. This is something that you have to know. All right. So your H&E technique, this is an example of a regressive stain. Okay, why is it a regressive staining technique? Because it makes use of, or it incorporates a decolorization or differentiation step. Okay, apart from that, your H&E technique is also an example of counterstaining. Why? Because again, there is a counterstain that is incorporated in your H&E technique. So it is both a regressive and a counterstaining method. Okay, so in the boards, which one is a better answer? A better answer is your regressive staining. Okay, yes, in the boards, a better answer is regressive staining. Okay, so what are the different major steps in your H&E technique? So I will no longer include the duration of immersion of your tissue section because we will be following the one that is in your module 4, activity number 6. Tama ba ako? Yes, I think it's in activity number 6. So we will be following that. Okay? So yung duration na nandyan. Duration of immersion of the different sections in your H&E reagents. Okay? So I will be explaining lang the function of each reagent. All right. So what are the different major steps in H&E technique? First, it says there, okay, first it says there, you have to immerse it in 2 changes of silene. So what is, okay, what is the purpose of the 2 changes of silene in the first portion of your H&E technique? So this is for deparaffinization. Okay, so this is for deparaffinization. All right, ma'am, I already performed deparaffinization before the staining technique or before staining. It's not enough. Yes, you still have to remove the excess paraffin that is present on your tissue sections. Otherwise, okay, otherwise your paraffin will interfere with the staining of the different components of your tissue sections. Okay, so that's why deparaffinization, this deparaffinization step is incorporated in your H&E technique in order to further remove excess paraffin that is present in your section. Okay, yes, otherwise, again, your paraffin will interfere with the staining of your tissue components. All right. Another, we have descending grades of alcohol. Okay. So again, when we say descending grades of alcohol, we start from high concentrations until such time that you are able to go to your low concentrations. Okay. So what is, yes, what is the purpose of this set of reagents? So this serves for hydration. Okay. In some of the references that we have, the descending grades of alcohol, okay, this is otherwise, this step, is otherwise referred to as bringing slides to water. Okay, bringing slides to water or hydration. Why do you have to hydrate? Why do you have to bring your slides to water? This is because, okay, this is because, class, you have to make your tissue section compatible with the first stain that is incorporated in your H&E technique. What is the first stain that is incorporated? We have your hematoxylin. What solvent do we use in order to make your hematoxylin solution? Water. Okay, so therefore, your hematoxylin here, it's an aqueous. It's an aqueous solution. Okay, it is prepared using water. Okay, is your section, is it inherently compatible with water? No, it's not. So you have to hydrate. You have to make your tissue section compatible with the aqueous hematoxylin solution. Alright, ayan. So after hydration, you have to wash it in running water. Okay, yes. And then after that, you are going to immerse it in your hematoxylin. Okay, so this is another major step in your H&E technique. So what is the purpose of your hematoxylin? This serves for nuclear staining. So your hematoxylin class, okay, so this is actually regarded as the primary stain in your H&E technique. Okay, so it is for nuclear staining. It is the primary stain in your H&E technique. Okay? Yes. So after that, before that pala, so for your H&E technique, there are two kinds of hematoxylin which may be utilized. Okay, so we have your Harris hematoxylin. Okay, Harris hematoxylin. And the other one is your Ehrlich's hematoxylin. Okay. So for your Harris hematoxylin, immersion, the duration of immersion should be 5 minutes. Okay. For your Ehrlich's hematoxylin, the duration of immersion should be 15 to 30 minutes. Okay. Yes. So between the two, which one is preferred? Of course, it's Harris hematoxylin. Why? Because of the less time. Okay. The one that is incorporated in your module, that is actually your Harris hematoxylin. Why? Because it requires a shorter time for immersion. Okay, shorter time of immersion. Okay, ayan. So we have two hematoxylins. Okay, so after your hematoxylin, you are going to wash it again in running water. And then after that, okay, and then after that, you are going to immerse it in 1% acid alcohol. Okay, so how do we prepare your 1% acid alcohol? So 1% acid alcohol, this is prepared using 1 ml of concentrated hydrochloric acid plus 99, okay, plus 99 ml of 80%, okay, of 80% ethanol. Yan. So that's your acid alcohol. 1 ml of concentrated hydrochloric acid plus 99 ml of 80% ethanol. Okay? So your acid alcohol class, this functions as your decolorizer. Dito ko na lang lagay ha. Your acid alcohol, so this functions as a decolorizer. Okay? A decolorizer or a differentiator. Okay? Decolorizer or differentiator. Okay. So anyway, decolorizer or differentiator. So that's your 1% acid alcohol. Okay? Yes. So in the laboratory, so since your acid alcohol is a decolorizer, it is actually the one that we use in order to remove stains from the skin or on the skin. So say for example, you are quite messy, in the laboratory, especially during staining, and then you did not use gloves during the staining process, and then your hands, okay, your hands got so colorful with all of the stains, particularly your hematoxylin, or yeah, even for your eosin. So what can you use in order to remove those stains from your skin? You can make use of your decolorizer, which is your acid alcohol. Okay, so that is another use of acid alcohol in the laboratory, particularly in histopath. Okay, so after your acid alcohol, okay, just to refresh our knowledge on acid alcohol, remember that you have to immerse your tissue sections in your acid alcohol for a short duration, very fast. Ganun lang. Okay, ganun lang, best. It should be very fast. Why? Because your acid alcohol tends to wash out. Okay? It tends to wash out the tissue sections on your slide. So that's why a lot of my former students before, okay, yes, a lot of their sections got washed out by the acid alcohol. Yes, you have to perform it very fast. Ganun lang dapat. Okay, yes. Otherwise, your sections will be washed out. Okay, yes. All right. Ayan. So, after your acid alcohol, you have to rinse it again in tap water. And then you have to immerse it in either your ammonia water or your 1% lithium carbonate. Okay, so what is the purpose of your ammonia water, okay, of your ammonia water, or your, okay, or your 1% lithium carbonate. So this functions for bluing. Okay, so that means it enhances, okay, the color that is produced by your primary stain, and that is your hematoxylin. All right, so that's bluing. Okay, bluing po ang tawag dyan. So, ammonia water and 1% 1% lithium carbonate, these are otherwise referred to as bluing agents. Okay? So, apart from these bluing agents, we can also make use of other bluing agents such as warm tap water. Okay? So, we can make use of warm tap water. Yes, particularly the temperature that is required is 40 to 50 degrees Celsius. Okay, warm tap water. What else? You can also make use of bicarbonate. Okay, bicarbonate. You can also make use of potassium or acetate. Okay, potassium or sodium acetate. Okay, and then another, you can also make use of ammonia. Ay, hindi na kasha. Saan ko ilalagay, class? Ito na. Okay, dyan na lang. Alright. Other bluing agents. Okay. So, warm tap water. What else? Bicarbonate. Potassium or sodium acetate. Okay. Ammonia. You can also make use of ammonia. You can also make use of Scott's tap water. Okay, substitute. Okay, so Scott's tap water substitute. Scott's tap water substitute. Okay, so sometimes they call it as your Scott's EWS. Okay. And so these are other bluing agents. You can go back na lang to my yes. Yes, you can go back. Okay. Yes. And these are the other bluing agents that you can use, okay, for bluing or for enhancing the color that is produced by your hematoxylin. All right. So after bluing, the bluing process, so again, you have to wash it in running water, after which you immerse it in your 5% aqueous eosine. All right. So what is the function of your eosine? What is the function of your eosine? Your eosine serves as the secondary stain. Okay, so it serves as the secondary stain. And sometimes in other references, it is referred to as the cytoplasmic stain in your H&E. Okay, it also serves as the counter stain. Okay, secondary stain, counter stain, or the cytoplasmic stain in your H&E technique. Okay, so after that, you have to dehydrate again. Okay, you have to dehydrate again by immersing your tissue sample in ascending grades of alcohol. So after which, you have to immerse it again in your saline. Okay, yan. So you have to immerse it again in your saline. So ascending grades of alcohol, this is for dehydration. And then the 2 changes of saline that is incorporated, this is for clearing. So that means it serves to make your tissue section, your stained tissue section, okay, transparent or translucent. So apart from that, the 2 changes of silene that is incorporated in your H&E technique, okay, this also makes your stained tissue section compatible with the mounting medium. Okay, yes, it has to be compatible with your mounting medium. Because what did you do in the H&E technique? First, you hydrated. Okay, so what can you say about the nature of your tissue section? It is somewhat aqueous. It contains water molecules. Are water molecules compatible with your mounting medium? No. Okay, so you have to remove your water molecules by dehydrating it using, ascending grades of alcohol. Is your alcohol, is it compatible with your mounting medium? No, it's not compatible. Okay, so that's why after immersing it in ascending grades of alcohol, you have to immerse it in 2 changes of xylene to facilitate clearing, making your stained tissue section translucent or transparent, and making your stained tissue section compatible with the mounting medium. Okay, so therefore, what are the different steps in, major steps in H&E? Deparaffinization, hydration, nuclear staining, okay, differentiation or decolorization, bluing, counterstaining, dehydration, and clearing. Okay, so those are the major steps in H&E technique. Okay, so I hope you are able to understand your H&E technique. Yes, and the last slide na, don't worry, okay? You can sleep already after this. So expected staining results, you have to be familiar with this, especially those that are highlighted. Yes, because they are being asked in the board exam. Of course, I'm preparing you for the board exam. So that's why I am highlighting those that come out in the board exam. Okay, ayan. But of course, you have to memorize all of this because I might ask them in your quiz or your exam. Okay, so expected results. We have for the nucleus or nuclei of your cells, blue to blue-black. Cariosome is dark blue. Cytoplasm and proteins, we have pale pink. Okay, RBC, eosinophilic granules, keratin, bright orange. Basophilic cytoplasm, plasma cells, as well as your osteoblast, purplish pink. Cartilage, pink or light blue to dark blue, depending upon the structure of your cartilage. Okay, calcium and calcified bone, purplish blue. Decalcified bone matrix, collagen, osteoid, we have pink. Muscle fibers, deep pink. Okay, and again, another one that is being asked in the board exam, probably to know if you have experienced your H&E staining, because I'm not sure if it is in some references. Okay, what do we use now? Okay, what do we use in order to avoid the slide surface contact, okay, between your glass slides, or in order to avoid surface contacts between your glass slides. So in Tagalog, so that they will not dikit-dikit. They will not make dikit-dikit during the staining process. Okay, so the correct answer to that is your paper clips. Yes, your paper clips. We use it in order for us to, in order to avoid surface contacts between your glass slides. Okay, so I'm sorry, it's 1 hour and 20 minutes. But anyway, yes, you can sleep already. Okay, so the quiz for this video will be on Thursday. Okay, so it will be po on Thursday. Yes. Ayan. So I'll see you again on my next video. Bye. Bye bye. Bye bye. Bye bye. Bye bye. Bye.